So this is the 4th and 5th page in the document that you have. So the 4th page has the sliding filament theory or mechanism described kind of in a shorthand version. But it goes through and it talks about, where various things will occur, but it has you coloring different components. And then I've added color in my little blocks, which will correspond to the colors on the picture. And then I've circled the important letters that you see on there. So I'm going to move this to the side so you can see the picture and how it's been illustrated and colored. So, but on page 4 you see it says the sliding filament theory explains muscle contraction based on how muscle fibers, actin and myosin, slide against each other to generate tension in the overall muscle. So it says that step number 1, a muscle contraction starts in the brain where signals are sent along the motor neuron. Color the motor neuron yellow. So this is our motor neuron right here. This is actually the axon. I'm going to go ahead and add some pointers and then I'll come back in and label them. This is the axon of the motor neuron. This is the axon terminus of the motor neuron. So a signal gets sent from the brain, and that signal is in the form of an action potential or nerve impulse, whatever you want to call it. It will travel down the axon and reach the terminus. Then we're going to talk about events that will occur here as a result of that, but just labeling some things you see here initially. The round structures in here are, I've colored in gray because they asked us to do that in the previous page. It says to color the vesicles gray. So these little round structures are called synaptic vesicles. And the synaptic vesicles contain the neurotransmitter released here. And the neurotransmitter released at neuromuscular junctions is always acetylcholine. So acetylcholine has been colored in orange. Acetylcholine are the orange triangles that you see here. Okay, so my orange triangles represent acetylcholine, which is a neurotransmitter. And it's a neurotransmitter that we always release at what's referred to, again, as a neuromuscular junction. So it's a union between a neuron and a muscle. Okay, then that neurotransmitter will bind to the green structures you see here. So the green structures are called ACH or acetylcholine receptors, and they're bound to the sarcolemma of our skeletal muscle cell. I colored the sarcolemma in brown, and of course I use that kind of light green for the ACH receptors. The location where those receptors are found is referred to as the motor end plate. So the motor end plate is a portion of the sarcolemma that contains the ACH receptors. There's a little space right here that the acetylcholine is secreted into and then will diffuse across, and that little space is called the synaptic cleft. Okay, now once the acetylcholine is released into the synaptic cleft, it will diffuse across the cleft and bind to our ACH receptors. That will initiate the formation of an action potential along the sarcolemma. So that's what these purple arrows are representing. Those are an action potential that's generated across that sarcolemma, and then that will be traveled or transported rather down through what's referred to as the T-Tubule or transverse tubule. At letter C, number 2 is the action potential, letter C is the T-Tubule. It asks us to color it in green, so I've colored it in green. The T-Tubule is simply an extension of the sarcolemma that will deliver that action potential down into the sarcoplasm. Surrounding the T-tubules are organelles that we don't have labeled here. These organelles are called the Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum stores and releases calcium ions. So the calcium ions are colored in here in blue. In response to the action potential moving down that T-tubule, the sarcoplasmic reticulum will release calcium into the cell. Calcium will go to the sarcomere and bind to troponin. So this is a portion of a sarcomere right here. You probably recognize the myosin. So these would be myosin tails, and these would be heads, which they're calling or referring to in the on the previous page. They're referring to them as cross bridges. So remember that a cross bridge and a myosin head are the same thing. Here you see strings of actin, right? Troponin and tropomyosin are also associated with this actin, but they've not illustrated them in this picture. So calcium actually will bind to troponin. It will cause troponin to undergo a conformational shape change, which will then remove tropomyosin from the actin active sites or myosin binding sites. When that happens, the myosin heads are allowed to bind to the actin active sites to initiate the contraction by doing a power stroke. And when these heads power stroke, it will pull the actin in towards the center of the sarcomere, which is what this arrow is representing here at number 5. That's the sliding of the actin or thin myofilament across the thick myofilament or the myosin to shorten the sarcomeres. It brings those Z lines closer together and will ultimately close the H zone. So I'm going to elaborate on those phenomena in lecture. I do want to go ahead and add some labels to this picture for you real quickly. So I'm going to go ahead and stop or pause the video, label it, put it back under, and let you make sure that you get all the important labeling down on your document. So let me hit the pause button real quick.